Okay, so we just kind of finished up with talking about some of the major contributors to the structure of DNA and its discovery. And it was a crystal image that was produced by Rosalind Franklin in her X-ray crystallography that really gave us an image of what we were dealing with. And it's a beautiful image of a double helix molecule. Now we're going to get into the chemistry of that in just a few minutes. But you had to complete something called Chargaff's rule, and I'm hoping that you looked it up. So anytime that you saw adenine, you guys knew that there would always be thymine, and anytime you saw cytosine, there was going to always be guanine. So in other words, A's and T's, C's and G's work together and they bond together, and it has to be just this so. So I was going to look up, and word for word, Chargaff's rule states that DNA from any cell of all organisms should have a 1-to-1 ratio of pyrimidine and purine bases, and more specifically, that the amount of guanine should be equal to cytosine and the amount of adenine should be equal to thymine. I mean, I feel like this is really kind of common sense. Is that everything should be in a 1-to-1 ratio. Now we know, and are going to uncover some violations to this rule just a little bit later. But Chargaff's rule states that DNA from any cell of any organisms should have a 1-to-1 ratio, so that the numbers should equal each other. So I'm hoping that you filled out that chart and you got it all attached to your notebook and it's all looking good. Okay, so now we're going to skip ahead just a little bit. Well, no, I think the next paper that you have gets into the structure of DNA. So let me see what my next slide looks like, okay? Yeah, so let's talk about it. And this was some vocabulary terms that I kind of picked out, and I thought maybe we could look at some current events and issues concerning DNA. And it's like if you've seen anything in the newspaper or magazines here lately, there's a big push right now, of course, with looking at genetics and immunology. And of course, we talked a little bit about this with our project, How to Stop the Spread of Infectious Disease. But so let's go ahead and start labeling or start defining some of this stuff. So we've got nucleotide. So nucleotide, and I'm looking for the definition, the scientific definition. It says a compound consisting of a nucleoside linked to a phosphate group. Oh my goodness, it sounds terrible. Okay, so nucleotides form the basic structural unit of nucleic acids. Okay, so I'm going to write basic structural unit, okay, of DNA, and it's also of RNA too. These are going to fit under the premise of nucleic acids, which is where we find nucleotides. And I think you have an activity. It says label the DNA nucleotide, so you're going to have to do a little bit of cutting here. And there was a place for you to write the definition, and then it goes and it says there are this many different nitrogen bases that can be part of a DNA nucleotide. So of course there are going to be 4 nitrogenous bases or nitrogen bases, and they are adenine, thymine, guanine, and cytosine. That's with an S. I'm so sorry. Gosh, my handwriting is terrible. Okay, cytosine. The sugar that is found in DNA is called deoxyribose. And if you guys remember, ribose sugar is also the sugar, found in ATP. Remember, I tricked you and got you thinking it was glucose. All right, so deoxyribose, it's missing an oxygen, and then of course the nitrogenous bases, and then on the outside backbone is going to be that phosphate group. And so your ears probably perked up. You probably said, well, wait a minute. We've got phosphate groups associated with ATP. We've got the sugar ribose associated with ATP. And, of course, these beautiful nitrogenous bases. And it gives you a beautiful image down there at the bottom of how this fits in. So you should have like these little, and I gave you an extra one just in case you needed it, but it says nitrogen-based cutout. So go ahead and cut those out and glue those into each one of your nucleotides. Fill in the circle with phosphate. So this is the circle, and then the deoxyribose, of course, is going to be is going to look like the little house, and then of course the square is going to be the base. So go ahead and click pause and work your magic, and let's label this DNA nucleotide. Click pause now. Okay, so you should be finished by now with your labeling your DNA nucleotide. I wanted to get into the double helix because DNA's shape has to take on this this double helix characteristic because it's for stable conformation. You know, remember, this phosphate group's got lots of negative charge, and negative charges tend to repel each other. So if we maximize the distance between this negative charge, I'm going to be able to hold my DNA molecule together. But it's exquisite. It's absolutely beautiful that it arranges itself in this way. Now, we'll talk about DNA replication at length a little bit more towards the end of our section with DNA, because then we'll get more into proteins. But I wanted to discuss with you the differences between purines and pyrimidines. And so that's going to be the next handout that I believe that you have. And it kind of looks like, I don't know what it looks like a hand with like 4 fingers or whatever. And it's also got some boxes that you need to cut out. It's got some directions. So you may need to look at the differences between purines, and you'll need to look at the differences between the pyrimidines. And then it asks you about the adenine thymine bond, and then the cytosine guanine bond. So you're going to have to do some extensive cutting, follow the directions. So go ahead and mash pause, and go ahead and assemble your purines and pyrimidines. Okay, so you've already established your purines and your pyrimidines, and let me go ahead and get us an extra page here. Okay, so I got me an extra page, so a little bit more room writing, and I wanted to kind of like maybe share some information or check your understanding for the differences between the purines or the purine bases. So purine is okay, so it's colorless crystalline compound with basic properties forming uric acid on oxidation. Okay, I know that means nothing to you, but they include, all right, so purines, let's make sure you got this. So purines are going to be adenine, and I believe guanine is the other one. This is not correct. Somebody needs to let me know real quick. But I think it's adenine and guanine, which makes sense because these guys wouldn't bond together. And then we have, because they've got 6-membered and 5-membered nitrogens. And then we have the pyrimidines. Pyrimidines. I'm trying to be real good with my spelling here. Okay. Beautiful structure. Because I'm looking at a picture of them. Okay. All right. So hold on just a second. Okay, sorry about that. Had to kind of put it back together. All right. So we've got the pyrimidines are of course going to be thymine, and I didn't spell that right at all. My apologies. Okay. So we've got thymine. All right, and then we've got cytosine. Okay, cytosine. All right, cytosine. And when you look at this, it kind of makes sense why the bases kind of work this way. So purines will pair up with pyrimidines. So you never want purines together and you never want pyrimidines together. So I don't know, maybe that helps you remember. I don't know. And maybe it's just easy remembering A's and T's and then C's and G's bonding together. But there is an activity that you guys were was kind of exposing you to them. And so, of course, what's the difference between them? And I was looking at an image, and I was trying to maybe share a resource, a really cool resource, where it would be purines will be 6-membered, and a 5-membered nitrogen, a 6-membered and 5-membered nitrogen-containing ring. So this is 6-membered. And a 5-membered nitrogen-containing ring. Okay, so there are 2 of them together, and in the pyrimidines, they only have 1 ring, and it's a 6-membered nitrogen-containing ring. So if you come across their structures, that might be easy for you to remember too. Pyrimidines just have 1 ring, and the purines are going to have 2. So that should have helped you kind of figure out your adenine, thymine, and cytosine guanine bonds. Okay. Now for the next part of the lecture, the next series, we're going to be looking at DNA replication. And there's a couple of different layers in this, a couple of different things that I wanted you to do, just different approaches that I thought would be really helpful to help you in kind of maintaining the difference between the purines and the pyrimidines and A's and T's and C's and G's. So we're going to take a look at that in the next lecture part.